So we just put the pyrimidines over here, and I'll do the same thing with the purines. I'm going to make them bigger and obnoxious, so we know what they are. Just increasing the size, and then I'm going to rotate them too. This little box, this little top right here rotates them. I'm just going to put them over here. So we have the pyrimidines. These are the single ringed nitrogenous bases. Don't forget, these are carbons, except for where there's nitrogens. And of course we have the purines, the double ringed nitrogenous base of the single nucleotides. Okay, now these are 4 different nucleotides, and it's interesting, all the 20 or so amino acids, and they're almost amazingly incomprehensible combinations of them in a huge 800 amino acid protein gives it its shape. But all the amazing proteins and the different combinations of shapes all come down to just 4 words in the DNA code. 4 expressions. And these are the codes that you guys have been talking about in 6th grade or 7th grade. Whenever you did this, okay, you talked about A, T, G, and C. Well, that's what these are. These are those A, G, T, and C. Let me show you. Okay, so, all right, the first one, insert a text box. Put this one right on top. We're going to name this one. And it's thymine. T-H-Y-M-I-N-E. And you feel free to make that bigger. I will. Okay, and make the box bigger. And there's thymine. So this is thymine. Now, specifically what makes this thymine though, because the phosphate and the deoxyribose are the same for the 4 different nucleotides. So really what makes this thymine is this particular nitrogenous base. So I'm going to put a big T in the middle. Okay? So I'm going to put a big T in the middle. All right? So I'm going to insert a text box. I'm going to make it a big this time. And I'm going to type in a T. I'm going to kind of select that T and make it really, really big. And I'll make it blue. Text color is right here. So it stands out. And then I'm just going to use my arrows. Oh, I got to select the box. I'm going to put that T right in the middle, the best that I can. And all I did was put a big T there to remind me that that is my thymine. Okay? And we put the T there because that's the T that we talk about in the code. It's not just a letter, it's really a 3-dimensional, okay, organic part of a nucleotide. That is the code. All right? And of course, thymine is a pyrimidine. All right. So I'm going to select thymine, and I'm going to go Command C, come over here somewhere, do Command V. That's basically cut and paste. I know it's not thymine, but I'm going to bring it over here just to save time. And now I'm just going to go backwards, delete it, and type in the next one. This one is adenine. A-D-E-N-I-N-E. And if you want to make it even, you can. And this is the name of this nitrogenous base. Of course, it's the name of the nucleotide, but what's really different is just this. So we're going to put a big A in this ring to remind us that that's the A part of the code. So to do that, I'm going to take this T, I select it, I do Command C, that's copy, then Command V, paste. I'm going to bring it over here, and I'm just going to go in that box, delete it, and write A. That way I'm not starting from scratch every time. And anytime we use the A like you did for your form, okay, those letters are really this. That's the link now. It's not just letters in an alphabet, okay? It's this, okay? And of course, A is a purine. Okay, selecting thymine again. Command C to copy. Command V to paste. Drag that box. I'm going to name this one. It's not thymine. go into the box, delete it. I'm going to hit cytosine, sight unseen, cytosine. And that's the name of this nucleotide. And of course we want to write a C, so I'm going to click the T, Command C, Command V. You certainly can go from scratch. I'm going to drag that copy, use my arrow keys that go in the middle. I'm going to delete the T and put a C. And last, hey, I'll click on cytosine. I'll do Command C, Command V. Bring that over to name the next one. Then I'll delete. Instead of this is guanine, Guanine. And of course, I'm going to put that G in one of the rings of that nitrogenous base. I'm going to copy the C, Command V, cut, paste. Hit the arrow keys, move the C there. Now change the C to a G. And so there's now life to your code, as opposed to GACD, which we'll be using a lot. What it really represents is this actual molecule in the code. Okay? Now you may say, Mr. Grodsky, why do we have purines and pyrimidines? Well, I don't know the reason for that, but I do know this. It is the nitrogenous bases that link up by hydrogen bond. This electron deficient H can H bond to this O. What I want to tell you is that what links the inside of the code together is H bonding. And so these things are on the inside. I've drawn them all the same way for a reason, but this would flip over. What I can tell you is that a double-ringed purine is going to hook up with its partner that's a single ring. It's always a double to single. That keeps the what? Length of DNA constant. We're never going to have a double and a single. If you had those different sizes, DNA couldn't stay consistently the same width. So the proper pairing, as you guys have learned, is an A goes with a T. And I always remembered apple goes in the tree. And a G goes with the C. A car goes in the garage. And of course, if you have some other mnemonic that works for you, that's fine. But if you can see for a second, yeah.
For the adenine, if that deficient H, why couldn't it pair to the O of the cytosine?
Well, this could never do that because this is a carbon and hydrogen sharing electrons right down the middle. That's not positive enough.
No, I was the one above it.
This one can hook up with this O?
The cytosine O.
The cytosine O. Oh, it probably can, but it will not be as strong. I'm gonna give you a hint why it doesn't work as well. It can, and that's why mutations can occur in your code. But yes?
It probably won't work because of the, on the bottom of the cytosine, the N with the electron deficient H's.
Right, well, you're onto something. There is 2 places for H bonding occur when these guys push together. These guys get 2 H bonds. This one, okay, you've got this H, and someone else's H. There's 3 places that this can H bond. So so this was going to want to be, this is a stronger connection, more stable. I don't know if I'm showing that. Okay, I think I might have these flipped. But in any case, okay, so that's impertinent. Let's continue. Now it's going to get crazy. Okay, now I have purposely put adenine with thymine. Your next slide has the same pictures, just not in color. Okay, so it's exactly the same. All right, so we're going to start flipping these guys. All right, and I'll show you what I mean. So this is the same exact thing. All right, now what we're going to start doing is putting these guys together. All right. Oh, I know why I didn't flip it, because I have to flip the other one. Okay, cool. All right. So, as I said before, DNA, we know, sits in a double helix. Okay, of course, that was discovered by Watson and Crick, right? No, what's her name?
The Rosalind.
Rosalind.
Franklin.
Rosalind Franklin discovered it, not Watson Crick. Okay.
No, you just said it, so I just trusted it.
No, don't trust me. Okay, it was a woman who was not part of the man's community found out the answer. She was independently thinking. That's why we need more women in science. Guys get all testosterone-y about stuff. Okay, we need more women in science, okay? Any case. So, based on a double helix, for it to work, nitrogenous bases went inside. So we're gonna flip the first one. Watch how I do this, okay? So I'm gonna grab this, I'm gonna move it over. Now there's probably a better way to do this, and people always
I don't think there is.
rotate. Oh, here we go, flip
Oh.
horizontally. So now hold on to your ponies. The reason why I want to flip this horizontally is that we know that nitrogenous bases are the code that sticks together. Okay, it's a double helix. It's an anti-parallel code. So I want to flip this over to bring this nitrogenous base. And by the way, this is thymine, tree. This is adenine. These are complementary. This is the proper pair. So I want to flip that horizontal. So I'm going to click on it. I'm going to right click, and I'm going to rotate, flip horizontally. And there we go. And I'm going to bring this over. Now, we're going to number these carbons. Okay? Okay, so make sure I do this right for you guys. We're going to number these carbons. All right, so we're going to insert a text box, and that text box will be the same. Put a nice blue number here, bold number. I'm going to do number 1. I'm going to make it a little bigger, boop, 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 boop. And make it blue. Yeah, I like the numbers, and make it blue. Whatever. If you don't want to use blue, hey, I can't make you. All right. All right. Now, when we number the carbons, remember these corners are carbons. We always start numbering 1. There's an O here, it's not a carbon. We number the 1. This is the carbon that's attached to nitrogenous base. That goes 1 right there. That's the first carbon. Again, you say, Mr. Grotsky, where's the carbon? Because these things have so many carbons, they're making it simplified. The corners of the carbons. So we start right here. This is the second carbon. So I'm going to click on the number 1. I'm going to Command-C it, Command-V it to copy and paste. I'm going to bring it down here. And I'm going to change that, of course, click on it. Christmas, don't do that. Back arrow. Get inside the box first, there we go. And then type in 2. And you can see this will be the 3rd carbon. This is the 4th carbon, the 5th carbon. So let's keep going. These numbers are absolutely important to understanding the flow of this code. Remember, the code represents amino acids. This is 3. Okay. Command-C, Command-V. This is the 4th carbon. Inside that box, top 4. And then this is the 5th carbon, that's C up there. Always the carbon, other side of the O, attached to nitrogenous base. That, my friends, is the first carbon. And we number this way. Okay. Now, if you remember, oh, Christmas. If you remember your code, okay, from the homework a few nights ago, all right? Remember I asked you to write a code that was similar, that was complementary? Remember it was like 5 prime, A, T, G, C, and it went to 3 prime.
Yeah.
Okay, and hey, this is 5 prime, This must be what? 3 prime. The code runs anti-parallel to each other. They go in opposite directions. A goes to what?
T.
T goes to?
A.
G goes to?
C.
C goes to?
D.
Okay, and this would be the 5 prime end. So what is that 5 and 3? Here it is. The 5 is up. The 3 is down. So this one goes the same way. So this is the 5 prime ending. The other guy must be the 3 prime ending. So this has got, oh, that's the 5. This has got to be flipped. I've got to flip this one. Right? Where? This is the first, second, third carbon. If that's the 5 prime up, the other one's got to show the 3 up. This has to be upside down. So here's what I do with this one. I click it, right click it. We're going to rotate it. We're going to flip it vertically. Cool. Get these out of the way. Now I'm going to push these together. And maybe you can see what's going to happen here.
Oh, I thought that.
Oh, if you look carefully, this H will attract this O. So what I'm going to do here is put a line. Okay, so here's my shapes. Can I put a line? Insert.
Oh wait, no, it's right next to it, just line.
Oh, okay, yeah. Now I want that line to be dotted. Or can I do that? Yeah, I'm just put a line here between the O and the H. I can do this. And what I'm going to do to it is I'm going to check this dots. And make it bigger. You can see it. Again, just like Will told me, I'm going to put a line between the H and the N, because that is what? Electron deficient H's, right?
Oh, how'd you get the dotted line?
Oh, once I have the line, I click on it, I go up here, line dash.
Oh.
And because we don't see it, make it thicker.
Wait, how'd you get the line?
I did hit the line, dragged it, then I clicked, so I'll do that again right here. Like for instance, here's a bigger line. Here's a line. Okay, I'll make it bigger. Line weight 3. I click on it. And then right up here is the line dash. Some people say I'm dashing. And there's the dashes. I'm going to bring those dashes in between here. Oh, okay. And what do I have? I got a mistake.
Uh-oh.
Can this H bond to a C, H bond to this O? No, I gotta get rid of that. No H bonding there. So I click on it, delete it. What am I showing? H bonding between the what? Base pairs. All right, now hold on. I have one more thing to do. We need to connect this one here. And you'll be all set for tonight's homework. Okay, hold on. How do I connect this one? Well, I gotta flip it because it's got to go the wrong way. So I'm gonna go right click. I'm gonna go rotate, flip horizontally. Cool, get this one out of the way. Party people, what's gonna happen, all right, is, oh, that H. Ooh, I figured something was wrong. So this is gonna come, so this comes together. Can you guess that 2 H's and an O come out? Oh, dehydration synthesis. So water comes out. Another example. And what you get is an O there. Now what I'm going to do to get rid of this H is I'm going to go insert, let's go, hold on. Let's go insert a, oh no, let's go shape. Hold on, a little box here. Give me a little box. This little box is going to hide this H. Well, it didn't. Unless you do what? Fill it with what? White. It'll be all white. Make the box bigger. Now it's outlined, so I'm gonna make the line part of the box also white. Move it over, maybe. Okay, you get the point. And one last thing. This is a phosphodiester bond right here. Okay, we took out water. I'm gonna name it. You need to know it. Insert text box. Phosphodiester bond. This, my friend, diester. Diester. Yes, I couldn't spell, sorry. I'll make it bigger and phosphodiester bond. And that bond, I'll put a little arrow there, but that bond is right here. This is what links one nucleotide to another. By the way, this is, what's my next sheet? That's thymine. Put a T here, I know we're running out of time. Cytosine. Okay, so if I was doing this, it'd be what? Thymine, cytosine. What would be the complementary pair going the other way?
Oh, A.
Yeah.
Oh.
AG. That's how it works. 5' is up this end, 3' is up. We're at a good spot. I'm going to pick you up with a video that takes you from here to another position. Those that are here today, okay, you're also leaving with this. Hold on to your ponies for a second. There we go. Bunch of these. You guys know your 3 base pairs. Know your 2 amino acids that started your chain. You wrote those down.
Yeah.
Okay. Will, here you go. Sorry to keep you guys. Gonna cut these out, and you're gonna put 6 nucleotides together that code for. Give me the extras back. All right. I think you have enough.
We'll just tuck off.
Yeah, it's okay. I know. Well, I held you for a little bit. Sorry. If you need a pass, I'll write one.